So this is a genetics of yeast secretion pathway question. So when we think about the secretory pathway, we recognize that the signal recognition particle deposits proteins off the ribosome into the rough ER. And as the vesicles break off from the rough ER, form the cis Golgi, the cargo is transported through the trans Golgi into a secretory vesicle. And there it might end up at the membrane. So we know that yeast exists in haploid and diploid states, and we know that Sheckman used temperature-sensitive mutants to actually analyze the order of events in the secretory pathway. And what he did was he used epistatic experiments of yeast. And so here we see a table. You should take a look at the table pretty closely. So basically what happens is that we're generating diploid mutants by crosses here. And table 2 shows us the phenotype of each mutant. For example, SEC1-1 is vesicular, SEC12-4 is endoplasmic reticulum phenotype, and SEC14-3 is what's known as a Berkeley body. And the idea with epistasis is that we're supposed to order the event. So the secretory apparatus has a directional flow in the pathway. So when we think of epistasis, I like to think of baldness and brown hair. And that is if the gene that regulates hair growth, that is whether you're bald or not, is not expressed, then you'll never have brown hair expressed. So that is baldness is epistatic to brown hair. So if you don't have brown hair, it might be because you don't express the genes for hair growth. So an analogous thing can be done with the components in the secretory pathway with the ER, the vesicles, and the Berkeley body phenotype. So when we take a look at this table further, we're going to be able to answer this question right here. And the question is, the epistasis experiments are most consistent with the following directional flow of proteins through the pathway. And here you're given a bunch of choices. And so the first thing to do with this table is to recognize there's 8 alleles, but only 3 phenotypes, okay? And as you further take a look at it, what you're gonna recognize is that anything that has an ER phenotype, whether it's crossed to any of the other ones, it always gives you an ER phenotype, which means it's like dominant. So I think of like rock, scissors, paper. ER can kill anything. So that means that in the epistasis experiment, that in order to express the Berkeley body and vesicle phenotype, you have to have a functional ER phenotype. So what that tells us is that ER is the first one, which means that we can eliminate choices B and D. And so now it's between Berkeley bodies and vesicles. So we play the same game if we take the Berkeley body phenotype in SEC7-1 and cross it to anything. With the exception of ER, the Berkeley body is always dominant. That is, that you'll never see the actual vesicle phenotype in a Berkeley body cross. So it tells us that the answer must be choice C. So come check us out at medpathway.com. We're the MCAT experts.